great pleasure that Eric Foster is going to kick things off. Oh wait, I even had a slide for this. So this is where you can do the sign up. So if you didn't, sorry, I didn't know I had more slides like that. So just again, sign up, do this, look at this link. I mean, pay attention to Eric, but do this link and sign up for people you want to meet with. It's like great. Oh, there's the links. Okay. All right. All right. Now let Eric.
How's that? Good?
Yeah, it's great.
Awesome. Oh, that's like huge relief to me. Because I hate, standing right over the room. So now I'm free to walk around. Okay. So I'm Eric. My name's Eric Foster. I'm an assistant professor here at the University of Iowa for biostatistics, working in the Clinical Trials Center. You've all seen Chris Coffey, who's the director of the center. Got up last night, told you all a few things you could do in Iowa City if you had a chance. One thing he left out, unfortunately, was, and this is kind of unfortunate, the lone president that Iowa lays claim to is Herbert Hoover. And so yeah, the president that presided over the Great Depression. But there is this awesome presidential library, not library, museum, which is kind of rare. There's a lot of libraries, very few presidential museums, just outside of town, a 15-minute drive, if you get a chance. And I always like to say it's like, it tells me about this much of his past and this much of his presidency. Okay. That's actually not a bad segue into this talk. Because we'll talk about clinical trials history and some of the history behind regulations that we have to deal with nowadays. First, I don't think I have to sell anyone in this room on the importance of clinical trials, but whether or not you're involved in clinical trials, they're a big part of our daily lives. So it's not uncommon to open up a web browser, go to a news site, and then right there is a story that has come about due to the results of a clinical trial. For example, when I was revising the slides, I went to CNN.com, and boom, right there at the front page, there's a story about the new immunotherapy and cancer, and the FDA looking to approve this immunotherapy based off of a phase 2 trial. So it's actually kind of interesting. But anyways, not uncommon to see clinical trials in our daily lives. It's actually not really anything new. I mean, health has always been a concern. We can go back as far as we can in recorded history and find observations and studies on health. For example, King Nebuchadnezzar in his court had an experiment where he divided his court into a group that ate meat and drank wine every day, and then a group that was more like a vegetarian group. And looked to see how they were doing over time. So clinical trials actually has clinical studies, I should say, actually have a really rich history. Sometimes it's not so good, like if you think about classical Greece, actually the reverse clinical trial where you inflict someone with the condition and then try to treat them. But I think what we'll see through this talk through the history of clinical trials is we'll actually see that a lot of the things that we do today are surprisingly recent. Okay? So I think what we'll start with is one of the first recorded instances of an unintended clinical trial. So this is Ambrose Paré. I'll probably butcher that name. It's in French, 1537. He was a barber surgeon. So it's back when physicians wouldn't be the one who operated on you. You would go to a barber to have surgery. And so he would often accompany the king's men during a battle in case surgeries were needed, or for any sort of healing that was needed after the battle. So his population was a group of soldiers with wounds sustained in a battle. The standard treatment of the time was boiling oil. So if they had a wound, you would pour boiling oil on the wound. The test treatment, basically what happened was there were so many wounds, they ran out of boiling oil. And so he was actually aware of sort of an ancient treatment at the time. It was a Roman treatment, a combination of egg yolks, oil, roses, and turpentine. And so he applied that after they ran out of boiling oil. So the soldiers treated with the test treatment slept through the night and felt little pain. And those treated with boiling oil were feverish, with pain, and swelling around the wounds. And so he's quoted as saying, "I determined never again to burn thus so cruelly the poor wounded by arquebuses." Does anyone know the arquebuses? Any guesses? No? It's actually like an old, really old version of a gun. They used to have to like put it on a tripod and everything. Anyways, he had this accidental experiment, and it changed the way he treated people back. We fast forward 200 years. We come to something that's a little bit more well known: Lind's scurvy study. So Dr. Lind was a physician on a boat called HMS Salisbury. And this is sometimes considered the first recorded controlled clinical trial. And so the population here was 12 British sailors that had scurvy. The control treatment, the standard of care at the time was either seawater or fresh air. So the control treatment in his trial was seawater. And the test treatments were going to be cider, elixir vitriol, vinegar, nutmeg, and oranges and lemons. Okay, so we all know today scurvy is a vitamin C deficiency, right? So it's obvious that oranges and lemons are going to work. They had no idea about this at the time. He put two sailors on each of the treatments. And so after about 6 days, the 2 sailors that were on oranges and lemons were up and about and working. So it's drastic improvement compared to the other groups. Now, what I love about this example is what happens after that. Because I think it mirrors things that still happen a lot today. What happened was Dr. Lind himself, and even after conducting the experiment and seeing the results, still recommended as the first treatment fresh air. Okay? So obviously seawater didn't work, but fresh air probably still does. And the next thing that happened is it took about 50 years for the Navy to adopt the citrus diet. So evidence starts building, and it takes time for it to be adopted by the whole culture. And then finally, and this is kind of funny, the Navy, even at that point when they adopted the citrus diet, oranges and lemons, found it was too expensive, and so they switched to limes. And so they found like they're generic or whatnot. So it's a really apt example. Okay, fast forward another 100 years, and we get to Flint's rheumatism study. So this is often considered the first placebo-controlled trial. And it's in a population of inmates, 13, and the treatment was mint water. So it's kind of like, you've probably heard of historical controls, right? This is almost like the historical active treatment and the concurrent control. So he's recruiting subjects into the study, giving them placebo. And so what he found is when he gave them mint water, they had the same positive results as were shown in other studies that had just the active treatment. And so the suggestion from this trial was that too much importance was being placed on giving a medicine, and not enough importance was being placed on the body's natural tendency to heal. Next up, we go to about 1943. British trial, so the MRC, it was the Patulin trial. This is the first double-blind clinical trial. It was in office workers and factory workers, and it was a Patulin versus placebo. And they were trying to treat the common cold. And the allocation here was an alternation procedure. So the first person in gets one treatment, second person gets the other, third gets the first, fourth gets the second, that sort of thing. So it wasn't randomized yet. Okay? The summary was it didn't show any protective effect of Patulin, but they were successful in giving the treatment to patients without the patients knowing what they were getting and without the physician necessarily knowing what they were getting. So it was a double-blind clinical trial. It's often overlooked because it was a negative study. Still successful, it answered its question. It was a negative study. More on it later. Okay, in 1946, just a few years later, we got a big development. It's the first randomized clinical trial. This one is a more famous study, the streptomycin study. It was in patients with pulmonary tuberculosis, and the treatments were streptomycin and bed rest versus bed rest alone. Bed rest alone would be the placebo. The allocation was a randomization procedure using random sampling and sealed envelopes. So they used a random number generator to generate the sequence, put everything in sealed envelopes, and only when you open up the next envelope would you know what treatment the patient was going to get. So it's a true randomization. The summary was it demonstrated that streptomycin is beneficial, but patients did end up showing resistance to the drug. Okay, so we've walked through some of the key developments of things that we think about a lot today, double blinds, randomization. When did those first start appearing in clinical trials? Next, let's talk about some of the regulations that we need to think about when we're putting together our clinical trials. During a lot of the time spent spanning what we just saw, there really was no regulation. And so you end up with things like these ads from the early 1900s. Like the top left here, cocaine tooth drops for kids. The one on the top right here is a pretty famous example, Hamlin's Wizard Oil. It was an entire circus act that would literally come to your town promoting a drug and trying to get everyone to buy this drug to cure tons of different things. So it was really, I guess you could call it the Wild West of advertising and drug manufacturing and selling. Now, with regulations, what we're going to see is a pretty consistent pattern. You start out with none, and then you wait till something bad happens, and then you react to it. And that's sort of the path of regulation, especially in clinical trials. So 1906, the Food and Drug Act. This was in response to the deaths of several children due to contaminated smallpox vaccines. So terrible tragedy struck the nation. This was the sort of government response to it. Now what this did is it provided a legal definition for the terms adulterated and misbranded. So there's still no drug review. And there really isn't an FDA at this point. I think it was like the Department of Chemistry still at this point. Basically you're only required to meet certain standards of strength and purity, and there's no requirement to submit to this FDA-like organization. And so basically, all they're able to do is say, if you say X, Y, and Z is in the bottle, then X, Y, and Z needs to be in the bottle. That's basically it right there. This led to a big Supreme Court case, 1911. U.S. versus Johnson. The government has seized a large quantity of product called Dr. Johnson's mild combination treatment for cancer. This is actually an advertisement for that treatment. The case went all the way to the Supreme Court, and the Supreme Court actually sided with Dr. Johnson. And what the Supreme Court said is that the new law did not prohibit false claims, only false and misleading claims regarding ingredients or identity of the drug. So basically what they were saying is that therapeutic claims are subjective, they're opinion, okay? If you think cancer, this thing cures cancer, then maybe it cures cancer. So what this actually did is it led to the Shirley Amendment, which prohibited labeling medicines with false therapeutic claims. So that meant like on the bottle, not necessarily in an advertisement. But this is a big one. The government had to prove intent to defraud. So super tough for the government to make a case. Okay, next tragedy. 1937, elixir sulfanilamide disaster. Sulfanilamide was one of the first wonder drugs used to treat strep throat and streptococcal infections. It was in response to consumer demands that the company made a liquid form. And that's the solvent used in that liquid form was diethyl glycol, which is a poison that's related to antifreeze. And the product was not tested at all in animals or humans before they marketed it. Okay, so they just made this new formulation, sent it out. This led to deaths of more than 100 in the US, and that was predominantly in children. Okay, so it's another tragedy. Next year, we have the Food, Drug, and Cosmetics Act. The set of laws giving authority to the FDA to oversee safety of foods, drugs, and cosmetics. Prohibited false claims of therapy and required drug sponsors to submit safety data to the FDA. So now you're actually having to interact with the FDA. Okay? So it used to be you didn't have to, now you do. You have to send them your safety data. The thing about this, though, is the default position of the FDA was approval. So the FDA basically had a 60-day window. You sent them your safety data. They had 60 days to block it. And then if they didn't block it, it was approved. Fast forward to the next tragedy. 1961, thalidomide. You probably heard of this one before, right? In the Billy Joel song, "We Didn't Start the Fire." Anyone? Maybe a good karaoke target. There's a lot of here. That's a tough one, yeah. You have to be a pro, maybe. The thalidomide was a popular sleeping drug in Europe. It was discovered to cause severe birth defects in babies whose mothers took the drug during pregnancy. It was never approved in the United States, but the drug sponsor had sent thousands of samples of it to doctors all across the country. And those doctors had given those samples to patients without telling them that it was experimental. And this unauthorized sample program led to more than a dozen thalidomide babies in the United States. So the next year, okay, here's the reaction, right? We get to see something that looks a lot more like the FDA that we know today. Basically, this amendment introduced a proof of efficacy. Okay, so no longer is it just safety and efficacy requirement. And you had to use adequate and well-controlled studies, clinical trials. The 60-day approval default was removed. This prohibited testing of drug in humans until preclinical studies had been done. It required drug advertising to disclose accurate information about the side effects. And it was the first U.S. law requiring informed consent. This is also where the IND becomes a big thing. Basically alerting regulators that you're going to be using this drug experimentally inside the United States, providing preliminary animal toxicity and information about manufacturing. Okay. Now there are a bunch of other things that have happened, right? This is just a few big key things along the way. You've probably heard of a lot of these, the Declaration of Helsinki, Belmont Report, things that we get trained on a lot when we go into health research. All the way up to, hasn't stopped 96, so we have ICH, guidelines for good clinical practice, clinicaltrials.gov requirements setting in, and then our CTSA programs. Okay, so that's sort of a broad sweep with the history of clinical trials, history of regulations. For the first talk, I guess it's appropriate to get into just some terminology. Okay, so clinical trial, definition of clinical trial. would be an experiment testing medical treatments on prevention strategies on human subjects. So an experiment, a series of observations under the control of the scientist. Clinical trial is prospective. Okay, we follow people forward in time. We try to recruit everyone at once, but everyone's got to have a baseline when you come into your study. And it's imperative there's an intervention. We're comparing different groups to one another. A randomized clinical trial is going to have at least one control treatment, at least one test treatment. We're going to have possibly blinding or masking. We're going to have to consider what type of treatment to use and what type of control to use. So, blinding, or in ophthalmology, masking, is the concealment of the identity of the intervention. Single blind refers to a patient not knowing what treatment they're getting. Double blind referring to when both the patient and the physician don't know what intervention is being given. Now, the more subjective the intervention, the more, it's not a specific number, it's sort of feel for what something is, the more subjective, the more important blinding becomes. In some cases, an unblinded trial is not possible. Like behavioral interventions or often surgery. Okay? And if blinding is not possible, perhaps the outcome assessment, being blinded, is possible. So having a third party assess the subjects. Now, treatments in clinical trials do not have to be drugs, right? We've already mentioned a few. It could be surgical procedures, medical devices, therapeutic modalities, diet interventions, behavioral interventions, exercise regimens, lots of different types of treatments for clinical trials. The type of controls, we've talked about placebo already. You can have active controls as well. You can add an agent to a standard regimen, and also different doses or intensities of the same intervention could be compared to one another. As far as the phases of the clinical trials, there are 4 phases of clinical trials. Phase 1, we're looking at safety and toxicities. It's a dose-finding phase, often first in humans, small, uncontrolled, healthy individuals. A lot of times it's a college-age kid, right? There's the famous example with Pfizer and what drug that would come to be known as Viagra, when they were doing their phase 1 studies, and they recruited a bunch of college-age kids and gave them basically the equivalent of a dose you give to an 80-year-old man. And we started to realize some of the side effects of this drug. So one of the things we'd like to get out of a phase 1 trial, maximum tolerated dose, or a range of doses that we'd like to carry forward into future research. Okay, phase 2 is our proof of concept period. Here we're examining whether the drug has sufficient biological activity. And here we tend to have, sometimes it seems a little counterintuitive, but we tend to have strict eligibility criteria here, because you want to focus in on a group where you really think this could work, okay? And the keynote address last night talked about ways of actually using adaptive trials to expand that a bit. But in a lot of phase 2 trials, I see some pretty strict eligibility criteria. They can be with or without comparison groups. They can be performed in patients with the disease now, like the phase 1. Here, a lot of times what we're trying to get to is it's called a go, no-go decision. So after this study, do you go on to a larger phase 3 study? Or don't you? So really the goal here is, quote unquote, killing trials early before you've invested a lot of resources in something that's not going to work. So figuring out with high probability, is this drug effective or isn't it? And also determining the profile, safety profile. And clinical trials are known to not be so great at determining safety profiles, at least through phase 1, phase 2, phase 3, especially like rare events that occur late, or chronic events. But there are things that are done to try and pick up on those. In phase 3, here's our confirmative evaluation of effectiveness, the overall benefit-to-risk ratio. Here we're comparing with maybe a standard therapy or placebo. You have a large sample size that's generally multi-site, and there's all sorts of hypotheses that can be tested in phase 3. Phase 4, this is our long-term surveillance studies. Post-marketing is what they're often referred to as. Here you continue to collect evidence regarding safety, efficacy, and toxicity of the treatment. And here is where you could find those rare events, those late onset events, maybe chronic side effects. And so here's where you get like a new warning label for a drug, or even a drug being pulled off the market. And then finally, this kind of different type of trial, comparative effectiveness trial, basically setting up studies of different interventions that are acceptable to use in treating a disease, and trying to come up with rules to inform healthcare decisions, perhaps based off of some type of covariate, a subgroup. Okay, what do you do in this subgroup? Do you treat them with A or B? That type of thing. Okay. So some key elements here to wrap up the talk. The fundamental point is that every clinical trial must have one or more primary questions of interest. They should be designed to answer those questions. Those questions should be answerable. There's the sample size is going to be generated off of this question, these questions. And one thing I wanted to point out was that there's a difference between a positive and negative clinical trial and a successful clinical trial. So a successful clinical trial answers the question that you went in to answer. Regardless of whether it's positive or negative, if it answered the question definitively, it's a successful clinical trial. Okay? Maybe not from the perspective of treating a disease. Okay? A negative clinical trial could be considered not a success. But from the perspective of performing the clinical trial, a negative study, okay, that definitively answers the question and says it doesn't work, is a successful clinical trial. Now, protocol, we're all developing protocols in this course. The protocol provides a comprehensive description of key information for conduct of the trial. It's the go-to resource of the trial. It should be adhered to as strictly as possible, which means it's got to be pretty darn detailed. Okay? And any deviations from that protocol should be reported. And here's just a brief look at sort of a table of contents for a protocol. You start with a synopsis, an overall look at your study, and then section 1 would be the study objectives. So we lay out primary, secondary, perhaps tertiary objectives. Background would be step 2. We have the rationale and supporting data. Explain to everyone why you think this is going to work. Step 3 will hold an overview of the study design. Part 4 is going to have the selection and enrollment of subjects. So this is really the inclusion, exclusion criteria, and how you're going to enroll your subjects. In step 5, you'll have the study medication, drug, and device material. This is where we've listed out interventions and how we're going to deal with study drug, perhaps. In part 6, we've got clinical and laboratory evaluations. So here you've got your schedule of events. This is, in my experience, this is a really high traffic area of the protocol. Just about everyone working on a protocol is going to look at this at some point. Management of adverse experiences is part 7. This is supposed to be part 8. The criteria for intervention discontinuation. In step 9 here, we've got statistical considerations, part I'm very familiar with. You have general design issues in there, outcome sample size, how you derive the sample size, assumptions that we're making about accrual of subjects, accrual rates, and anything we're doing about monitoring data. In part 10, we'll have data collection, site monitoring, adverse experience reporting, what records are being kept, do you have a data management team, what's their role, do you have a quality management team, what's their role, and adverse experience reporting. And in part 11, we've got our human subjects section. So here we're talking about the IRB review and informed consent, subject confidentiality, study modification, discontinuation. Part 12, publications of your research findings, and part 13, list of all the important references. Okay, I think that's all I've got as far as the introduction. If there's any questions, I'd be happy to answer them. Otherwise, welcome to Iowa.
I can talk about that.
Yeah, sure.
Thanks.
So the FDA, they certainly will look at what's been done outside the country.
Yeah.
If there's a large database, we do tons of device studies, for example, where the drug's been in Europe for years. They will use that. You can use that potentially as even historical controls in phase 3. So we've got examples, kind of a cool example where there's a 100,000 patient database in Europe, but it's not on the market here, and they've leveraged that. So you absolutely can. The degree to which you can use it, of course, is going to be a subject of negotiation each time. But if you're talking about safety profiles, you should absolutely, it's a point of discussion to have over how to use that and what, but it's on the table.
Eric.
Thanks, Rick.
Just to add to that, this is a moving target, and those of you with neuromuscular disease may know that the new FDA-approved drug for ALS that actually is available to patients today, that no human being in the United States has ever studied. All of the studies were done in Japan, from phase 1 to phase 3. They presented the brain for having completely in a population and inclusion. I don't think that would have happened 5 years ago, but just, I guess it goes to show that we're moving towards globalization.
Did you say this was a robot?
Yeah, it's Cyberman, the Japanese robot that helps with walking.
So there is a major difference between how the FDA treats drugs and devices. So, to a certain extent, the phase 1, 2, 3 isn't exactly salient to devices. Drugs, and correct me or anybody else, since there's a lot of knowledgeable people here, correct me if I'm wrong. Drugs need to be shown to be safe and effective. And typically this is exploratory phase trials, which would be maybe what we call phase 2, phase 1, etc. And then confirmatory trials, which typically we call phase 3. In devices need to be shown to be safe and to do what they say they're going to do. For example, mechanical thrombectomy devices, they need to be able to be shown to pull clot out of the brain. Now, recently, last night, you heard of recent clinical trials that showed that they were very effective in reducing stroke-related disability, which was great. But to get those devices on the market for a device, the FDA only knew that they could safely pull clots out of the brain. And then the clinical community wanted to know selecting the right patients for these devices, how to select the right patients for these devices. So the device is met with a different pathway than the drugs. And I don't know, does anybody want to add to that? Is that
I will add that the terms phase 1, phase 2, phase 3 are really drug development terms, and the equivalent in the device world is pilot and pivotal studies, and it's much looser, so it's hard to apply phase 1, 2, 3 to device studies.
Thanks.
All right, so our next talk, we're waiting on some folks who are going to come from the room next door. So I think we can just mingle and plan to reconvene like right at 9.
It's just a recently. How are you? But it's intermittent. Yeah, that's very important. I'm sorry. I'm satisfied.
Yes.
This one's like real like place for a long time.
Yeah, right.
Yeah, no, yeah. Right? Yeah. Your plan.
It's not like we think about it.
Exactly. So that's a that's just I'm trying to write my story. Yeah. Dark side. Yeah, it's tough because we're competing with this penalty is the margin. No, no, because I
Pull down. Yes. Yeah, it sounds good. I don't know what that is.
Wendy, before I forget, can we have dinner on Thursday night? Culture movies or whatever, yeah. Excuse me, we have just oh, okay, but I don't want to go out late. I just want like dinner. Just as short.
Okay, so I can
Good.